Welcome back, everyone, to our next session, Therapeutic Modalities and their Relevance to Your Disease. This session is going to explore the different treatment modalities and how a strong understanding of your disease biology, genetic etiology, prevalence, etc., can really help to inform the most appropriate therapeutic modality for a particular condition. Please join me in welcoming our panelists, Danny Levine, Daniel Fischer, Sarah Gladstone, and Tim Yu.
There you go. Can you hear me now? So we're going to talk about therapeutic modalities. If you think about the evolution of modalities, we've seen this movement from a throw it at the wall and see what hits approach to very targeted approaches from treating symptoms to cures, and this move towards precision medicine and now rather elegant approaches to targeting disease-causing genes. But with that in mind, we've got a panelist that can think about modalities in very different ways. We have an academic researcher, a pediatrician turned rare disease mom turned chief scientific officer of a rare disease organization, and we have an MBA engineer who is now not only a parent of a child with a rare disease, but also the CEO of a biotech. And I think what you'll see as they talk that they may each think about modalities in different ways. With that, I'm going to ask Tim Yu, who is our academic researcher, to just make sure we're all on the same page and start by explaining what is meant by the term modality.
Thank you, Danny. So I'll take first crack at this, and by way of background, I'm a neurologist and a geneticist at Boston Children's Hospital. It's been my privilege to work with the rare disease community since I started at Boston Children's in 2007. First on the discovery side, discovering rare diseases, and then most recently trying to develop therapies for them using increasingly precise approaches. So I'll just try to set the table for my colleagues to expand on. When we talk about modalities, what we're talking about is the particular tool that we are going to contemplate using to try to develop a strategy to treat a given genetic disease, rare or not. And we might start with the basics. We might start with small molecules, which are the most traditional type of drug that you will think about when you think about this term therapeutic modality. These are small chemical molecules that have been synthesized to have a very precise effect on the biology of a patient that can be useful for treating certain kinds of diseases, especially those where we know the biological role of the gene exquisitely well. The reason for that is that these are sort of precision biology approaches. And this is an approach where many rare diseases, for instance, those that overlap with common diseases that we understand the biology of, that we've invested a lot of money and scientific research into, there's often opportunities for fantastic overlap between the biology of a common disease and the biology of a new disease, a new genetic disease. And we can use, say, small molecule technology, which has been around for decades, to try to develop or sometimes repurpose old treatments, develop new treatments or repurpose old treatments for those conditions. We can expand to that toolkit. And as an example, in the 90s, there was a lot of interest and the birth of this idea of viral gene therapy, where we use a virus to replace a faulty gene that's broken in an individual's body. And we deliver it to cells where it needs to go using a variety of tricks, but usually taking something like the virus that gives you the common cold or other types of viruses to deliver that cargo. And that's a modality that we've been working on now for some 30 years. let's go back to 20 years. 20 years ago, we invented a concept of something called a more precise gene-targeted therapy called antisense oligonucleotides. This is an RNA targeting therapy, short snippets of chemically modified bits of RNA that we can synthesize in the laboratory, manufacture, and then deliver to patients. This is a type of modality that is even more precise. It can be precise to the level of the gene. It can even be precise to the level of a patient's mutation. Lastly, now bringing us really quickly to the current, in the last 5 years, we've been seeing even more precise molecules and strategies such as CRISPR editing. These are modalities that let you target and correct the effects of a single mutation at the single letter level. And these are being rolled out for diseases like sickle cell or beta thalassemia, or also even for very, very rare diseases like biochemical disorders, such as that exhibited by a famous infant that was born at the University of Pennsylvania this spring with an inborn error of metabolism. We were able to use a precise CRISPR edit to change one letter in that baby's genome to correct, hopefully in a durable way, the error that the child was unlucky enough to have been born with. Those are a couple of specific examples sort of rolled out over time. Each of them have different advantages and disadvantages in terms of how long they've been around, how much experience we have with them, how safe we think they are currently. Some of them are cutting-edge technologies that haven't yet made it into patients, like tRNA therapies. We'll hear about those. And they have advantages of having amazing, wondrous properties, but yet little FDA experience. So in thinking about each of these, you have to think, what can we do today? Who are the partners who are experts in this technology that I could partner with? What are the pros and cons in working with each of them? And also, how long will each one take to develop? And how much time do I have given the particular constituency that I care about, whether it's my child or my community, my disease community?
I think when most people start out on this path, they don't really care about modality or even think. They want a fix. Just give me something that works. Why should patient advocacy organizations involved in drug development even think about modalities?
I think that it's really critical to understand this landscape because this is rare drug development and drug development in general is not a linear path. It's very, very difficult to start at A with the expectation that you're going to get to Z. And unlike how science is portrayed in textbooks, it's rarely goes exactly the way that one wants. So in thinking about a strategy for your disease of interest, it's really important to have multiple shots on goal, to have multiple parallel irons in the fire, so to speak, because you need to spread your risk across multiple different strategies. And I think that that's a critical thing to appreciate. It's difficult because you need really well-seasoned science and pharmaceutical advisors to help you handicap each of these tools, but you want to try to balance the risk as you're thinking about how best to impact your community.
So Daniel, you're a biotech CEO. When you think about modalities come with all these implications about cost, manufacturability, and how it's delivered. As a biotech CEO, walk us through the process of the way, what are the issues or concerns that a CEO might think about in terms of whether they want to even get involved in pursuing a specific modality?
Yeah, so in my case, I have a double hat. My first hat as a father. That's why I started initially. As you said, I was looking for a fix. I have my daughter. She has a disease, Dravet syndrome. We know the type of mutation. It's a nonsense mutation. Anti-seizure medications don't address most of the issues. How can we address the root cause? Tim, you mentioned gene replacement. SCN1A is a very large gene, so we couldn't replace it. So we worked backwards with it. We reverse engineered, and we found a platform, a modality that will work eventually to solve the issue. That's the specific way that I started. We came up with the idea. When I talk about we, people who actually know about the science, my co-founders. But as a CEO, when I look at modalities, I basically look at two by two matrix. On one hand, I look at what's the probability of having an impact. What's the size of the impact that we can have with a modality? And when you think about impact, you look at the size of the patient population, you look at existing treatments, how good is the standard of care, and how transformative a new solution can be. So that's in one axis. And the other one, you look at probability of success. And when we think about probability of success, you think about how well the biology is understood, not only of the platform, but also of the disease. Can you deliver to the tissues this modality? How about delivery, which we know is a big issue, to the tissues of interest? How easy is it to recruit patients for this modality, for this therapy, et cetera? You look also, and that's a very important one for patient organizations, are there disease models, mice models available for the disease, cell line models? Is there a registry of patients that when we go to the clinic, we can recruit? And then what you basically map the different modalities. In an ideal world, you're gonna have several modalities that have high potential impact and low and high probability of success. And at the end, those are the ones that you go after, the ones that are gonna really increase your probability of getting to the clinic at a lower cost and a lower percentage time frame, and will be able to affect as many patients as possible.
So I want to talk about this in specific terms. You work in Wolfram syndrome, which is a complex neurological disorder. You know, can you start, since most people aren't going to be familiar with Wolfram, can you explain what it is and how it manifests itself and progresses?
Absolutely. And I think that it's actually a great moment to do that, because I'm listening to everything you're saying and going, oh my gosh, I never thought about any of that 10 years ago when I was looking at this disease. You taught me something already. I'm making notes in my head. So my son was diagnosed with Wolfram syndrome about 9 years ago. And the reason this is great timing is the only way I could think about how to treat this disease was to think, what's causing it, and what are the symptoms? So as a pediatrician, I have to simplify things and think about, well, I can either come at it from this direction, or I can come at it from that direction. So that's where I started. So we think about what are the symptoms of the disease. So this is it's a very complicated disease. When my son was diagnosed, I had trained right here, and I don't know that I ever saw a case of this in training. So it's an ultra-rare disease, but it's a disease that affects neurons and beta cells, the cells that create insulin in the body. And there's a reason for that we won't get into now. But the classic form, which is what I read as I got the diagnosis, was that kids will be diagnosed with insulin-dependent diabetes at age 5 or 6. They'll be blind by age 18. They'll have neurodegenerative symptoms and death between age 30 and 40. And so you can imagine my response at the time. There's a whole bunch of other stuff that goes along with it: hearing loss, diabetes insipidus, where you can't hold fluid in the body very well, GI dysfunction, bladder dysfunction, autonomic dysfunction, and my son calls it Diabetes Platinum Plus program. That was the name we came up with. At the time, there was a suggestion that it was a spectrum of disease, that not everybody had the classic form, and we knew at that time that we figured out my son had it by accident before there were any real symptoms of it, and that's a whole other story, but he was 11 at the time, so we knew something was different. And we now know there are hundreds of mutations, at least, that cause this disease. It may be recessive, it may be dominant, it will manifest in different ways. Huge spectrum of disease. So just to complicate a complicated disease further, we have to nail down every different way that it might present and find those people. So it may present in infancy, and I just had an email from a man a couple weeks ago who was diagnosed at 71. So how about that?
So, you know, when I started thinking this through, wanted to emphasize that thinking about modalities is a very three-dimensional exercise, but it's really four-dimensional because you have the element of time, and with progressive conditions, time becomes such an urgent issue. So you've got diabetes symptoms, you've got hearing loss, you have vision loss. How do you prioritize those symptoms in terms of getting a therapeutic strategy?
Yeah, that's the huge question.
Especially when you have all these underlying genetic causes that are
Right. So truly, when we got the diagnosis, the next morning, I was able to speak to our guru for Wolfram syndrome and say, okay, how do we fix this? Because on the phone, they said, well, there's no treatment when I got the genetic diagnosis. And I verbatim, I said, I'm really not comfortable with that answer. So at time, we put tremendous weight as far as time is concerned, because whether you have a progressive disease or a static disease, you're thinking about symptom progression, you're thinking about development of a child. Time in Wolfram syndrome, we say time is vision, because that's a symptom these kids really care about. They're not thinking ahead as much to respiratory depression and things like that happen with the brainstem degeneration, but time is vision. So we wanted treatment a few years ago would be great. So we have to use a three-pronged approach for this. This is something that just immediately became clear after I got to talk to a couple folks in the business, like you, that we had to think about slowing progression. We had to think about stopping progression. We had to think about replacing damaged tissue, and we had to do it all at once. So that's what we tried to do. And you're right, just like Tim said, we can slow progression more easily. We can come up with a molecule, a compound that may slow things down in many, many diseases. Stopping progression using techniques that didn't exist when I was in medical school, or replacing damaged tissue, also things that didn't exist back then, it's a little more risky, it's going to take more time.
So kids with Wolfram syndrome may have trouble swallowing. The glucose metabolism is off, so you have to be concerned about how a drug might interact with glucose metabolism. Urinary tract issues may have someone excreting a drug too rapidly. How do you think about modalities given the way that this disease might interact with drugs?
That's a great question. So we ask the kids and the adults now who have this disease. We actually just had a patient listening session with the FDA, which was fabulous, so that they could understand, and really we could understand where everybody's coming from. What's important to the patients? What symptoms do they have? I would say a lot of our kids and adults, I always call them the kids, they have swallowing difficulty. Some of them are young enough that a medicine that tastes really bad is just, they can't really tolerate it, although we find ways around that. Most of them are used to needles from being diabetic, so needle administration is not so bad. Glucose, so a medication we have in trial now had an undisclosed amount of carbohydrate in it. But the kids were all pretty willing to just work with that and figure out dosing for insulin as they took it and got a new regimen going. What we learned from most of the patients when we did this listening session and when we have conferences is that the folks with more severe disease, they would tolerate just about anything. That's what they say. We'll tolerate just about anything if we can come up with a cure or even something that meaningfully slows progression. So that anything from bad tasting medicine to spiking your blood sugar to intraocular injection to spinal tap, a lumbar injection of a medication to get to the central nervous system. They said very clearly we would do anything. Now, folks who have milder disease, they will say, yeah, I think I would do just about anything, depending on where I am in my disease course. But for sure, those folks say needles, bad taste for sure, blood glucose spiking, no problem. Intravitreal eye injections, yeah, no problem. So depending on severity of disease, that depends on what people will tolerate. And then depending on the details of a disease, the devil's always in the details, can that medication or treatment be administered depending on your disease, excretion, metabolism. Are there any really bad side effects you have to worry about? So risk-benefit ratio, severity of disease, these are all the things we have to take into account.
So, Tim, as you listen to all these symptoms, I'm sure the first thing that goes through most people's mind that want to get involved in drug development is, well, why are we going to treat these symptoms? Let's just cure the disease. And a one and done gene therapy seems far more seductive than going after all these different things. Why not do a one and done gene therapy?
I think that this is really, it speaks to how this concept of having a multi-pronged strategy is so important. I think that in terms of the hierarchy of things that you can do to improve the lives of folks with a genetic condition, managing the symptoms is the first and most proximate thing you should need to grab onto. And understanding what existing commonly used drugs may be useful for your patient population, targeting the specific symptoms that they have challenges with, learning from which of those commonly used drugs have been useful for other disease communities. That's obviously where every parent and every clinician needs to start. When you then think about, well, what new drug development can I do is necessary to fill the remaining gaps? And the remaining gaps after you've done the symptomatic relief might be huge, right? It's sometimes one finds that the existing drugs simply don't address the specific constellation that we're talking about. Well, the next level to reach to is repurposing of maybe less commonly used, but already approved drugs. And covering that literature, finding out what, and doing deeper investigation into what's been clinically used, maybe even looking at compounds that haven't been clinically used, but repurposing those in a screen of a model of your disease. And then from there, you can then progressively go towards higher and higher reaches. And you, Daniel, you prompted me with the pinnacle, which is the one and done gene treatment, right? The gene therapy, and whether that's a one and done gene replacement treatment, whether it's a one and done edit of your genome to fix the underlying problem. Those are absolutely the highest reaches that we go for in each of these situations. It's important to know that sometimes when you reach up really high, that you might have to incur some risk that you're not gonna get there, and you may have to build scaffolding, other alternative approaches that let you reach not quite for the one and done treatment, but maybe for the treatment that you have to give a couple of times a year to get the dose right, right? The challenge with the one and done treatment is that if you're giving it once, that often may mean that that one-time treatment could be a one-time overdose, or it could be a one-time underdose. And giving yourself the latitude to, and having the humility as a scientist to say that, well, we don't exactly know what the target dose is or the target time to treat is, may allow you to reach slightly less high and try a treatment strategy that requires multiple doses that can be given, say, a couple times a year, because maybe if you get the dose wrong in the initial doses, you can actually then step back and reduce the dose. So, oh, that was a bad idea. Let's halve the dose, or let's drop the dose by a quarter, or do the opposite. So each of these different approaches does have real scientific and clinical benefits to your strategy. Let me anchor this in an example. A lot of single gene disorders of the brain are now being treated, or are now the subject of drug development programs for one and done gene therapies. In parallel, many of those same diseases are the subject of drug development campaigns using, say, ASOs. An ASO is not a one and done therapy. It's something that you have to give once a month, once every 2 months, once every 3 months. Why would anyone ever do that? Well, if you could do a one and done therapy, well, the ASOs give you a chance to find the right dose, and they give you a chance to figure out how to measure benefit. They also give you a chance to figure out how big the benefit might be. And if you have that kind of information from an early first phase study, it can make the design of your one and done therapy trial that much more efficient.
You talked about ASOs, but we're getting a whole tool chest to alter anything from the gene itself to any step along the translation of that into a protein, whether it be an aberrant protein or a protein that's not expressed, what have you. To what extent does the actual genetic mutation define the appropriate approach, the appropriate modality?
The specific mutation matters a lot in your choice of strategy. And this is actually one of the real and most exciting opportunities that we have with this new scientific toolkit. By moving to precision genetically targeted therapies, we can now think of genetic diseases in a more unifying way than we were previously able to. It's one of the fundamental challenges of that the global genes community faces. The fact that there are 10,000 different genetic syndromes described. That kind of fragmentation makes it really important to try to find commonality. And if you can find this commonality by, say, taking those individuals within the community who have a particular type of mutation, and I'll use, if I may borrow an example from Daniel, if those individuals in the community who all have a nonsense mutation that you could treat with a tRNA that reverses the effects of that nonsense mutation, and I'll have to leave it to you to explain exactly more about that technology, but you can now convert this really highly fragmented community into a community that where you can develop a therapy that's actually relevant to many, many people within it in one stroke. Similarly, there are many diseases that might be caused by mutations that have a toxic gain of function effect. The mutant protein now is doing something that you don't want it to do. But if you can develop a therapeutic strategy that knocks down that gene, that toxic mutated gene, then there are lessons from that strategy that are cross-applicable to many other patients in the community. So that's the beauty of this type of precision therapy. It's that it is precise, yes, but sometimes precision can be a unifying force too, and it can allow you to group families and patients together under a common cause. And I think that that's the sort of the paradox of precision, that it's both on one hand very specific, but also allows us to change the way that we think about developing these drugs.
Well, Tim, you've been a pioneer in N-of-1 therapies. There are many people who take up drug development efforts because no pharmaceutical company is going to do it in the absence of not just de-risking, but just given the small population sizes. So to what extent do these therapies lend themselves to development for small populations?
I think that the challenge of ultra rare disease development really boils down to both bottom up and top down approaches. From the top down side, we need to try to make our rare diseases look really much more common than we currently appreciate them to be. We are currently under ascertaining the prevalence of these diseases hugely by not providing timely and equitable access to diagnosis. It takes much too long to achieve a diagnosis. If we really could diagnose everybody who is out there, we would know that they're much less rare than we think they are. And that's an important piece of the strategy. Using traditional drug development approaches, we can lower the threshold for which a disease might be commercially investable in that way. At the same time, we need bottom-up approaches too, which actually now allow us to develop treatments not on the basis, again, of necessarily just the individual disease, but to group them together in the way that we were referring to just now. And I think that that bottom-up approach will convert an ultra-rare condition into something which is not quite as rare. It's definitely novel, and it's a regulatory stretch, and it kind of challenges our notions of what a trial is. But if we can group as many of these strategies together and take common approaches towards treating them, and then come up with ways of regulating that approach, then we can unlock this completely different mode of treating kids. Very specifically, if we can treat one baby with a base editor targeting the liver to correct a specific biochemical deficit, then why can't we do that same thing for another specific biochemical deficit that just happens to be one gene over or a couple of genes over?
So Daniel, you work with tRNA. This is, to me, one of the more exciting areas of genetic medicine today because it has the promise of benefiting so many people. Can you explain what tRNA is and what nonsense mutations are?
So leave it to a non-scientist to explain the complicated science, but why not? Yeah, so in protein synthesis, basically you have a genetic code that's transcribed through transcription into mRNA. In the mRNA, you have triplets, and triplets are 3-letter codes that each one of them encodes for a specific amino acid. And the way it works is that in mRNA, you have the ribosome. Reading over the mRNA stops at each codon or triplet, reads the code, finds a tRNA that's floating somewhere around there, which has what's called an anticodon, which matches that specific 3-letter code, gets a tRNA releases an amino acid, and it starts forming a chain. And that chain, when it gets to the end of the mRNA, to what's called a termination codon, is released and becomes a protein. And that termination codon at the end is natural. Each message has a termination codon, and there are 3 different termination codons: a TGA, a UGA, and a UAA. Now, the problem arises when you have one letter, that's all it takes. One letter mutates in the message, in the DNA, and then the message. And what's supposed to be a normal codon or a sense codon that codes for an amino acid becomes a stop codon. And that's what we call a premature stop codon. It happens not at the end of the message, but it happens somewhere within the message. And what happens then when the ribosome gets to that premature termination codon, it cannot find a tRNA that reads the codon. Why? Because there aren't in humans any tRNAs that can read a termination codon. The reason is that when the ribosome gets to the end of the message with that stop codon, it should stop translation. So there aren't any tRNAs that can read that premature codon. So what happens is that from that allele, that gene, that mRNA, the system cannot create a full length of functional protein. So what you have at the end, it's a loss of function. You have a truncated protein, which is most of the time non-functional.
And what percentage of people with a rare genetic disease actually have a nonsense mutation?
Yeah, so that depends a lot, and it depends by disease. For instance, in Dravet, which is, of course, a disease that we started with, 25% of all cases have a nonsense mutation. If you think about other diseases that we're working on, in Duchenne muscular dystrophy, it's 15% of all cases. In other diseases, one that we're also working on, dilated cardiomyopathy, caused by mutations in the titin gene, it's about 40% of all cases are caused by nonsense mutations. So again, it depends by disease, but generally between 10 and 30%, some cases up to 40% of all cases are caused by nonsense mutations.
And just to clarify, theoretically, a single drug that corrects that could work across genetic mutations regardless of the underlying gene that's affected by a premature stop codon.
Yeah, so that's a great point. So at the end, a nonsense mutation is a nonsense mutation. It could happen in the gene that causes Dravet, SCN1A. It could happen in genes that cause other epilepsies like SYNGAP1, SCXBP1, CDKL5, SCN2A, et cetera. So in theory, you could think that one with the same drug, with the same tRNA that we're going to use to treat a TGA mutation We could use it for Dravet, we could use it for CDKL5, SCN2A, and others as well. So suddenly the idea of using one gene therapy to treat multiple diseases caused by nonsense mutation becomes extremely attractive. And of course, at the end, even N of ones, if it's caused by that same TGA or TA mutation, you could treat it with the same drug, same gene therapy.
Given that broad applicability, how do you as a drug developer think about prioritizing the indications you'll pursue?
Yeah, so again, going back to my 2x2, you look at potential impact versus probability of success. So we look at what's the size of the population, what's the addressable population, what percentage of the population of the disease is caused by nonsense mutations, how severe is a disease when it's caused by nonsense mutations, what other therapies are there in the clinic. For me, as I tell everybody, as a father, there are two things that I look at. One, is it transformational? Is it going to be transformational for the patient, or is it going to be a me too? I don't care. I don't care about me too's. I really need it to be transformational. With my daughter, for us, it was, is it going to treat the seizures only, or is it going to treat the disease as a whole? I mean, my daughter is 16 years old. She has the developmental stage of a probably 1.5-year-old. Non-mobile, is in a wheelchair, et cetera, et cetera. So for us, it's treating the disease as a whole. And the second aspect that I look at safety. We really have to make sure when we get something to patients that it's totally safe. So again, we really looked at one, it's going to help as many patients as possible, that it's going to have the ability to expand beyond what we call the core indication in epilepsy. Our core indication is Dravet, but can we expand it then into other epilepsies? In muscular dystrophy, our core is DMD. Can we then expand it into other muscular dystrophies as well? So the more patients that we can treat, the better. That's what we're looking for.
So one thing you didn't say in choosing indications, though, is your ability to target the right cells and tissues in the body. And I'll throw this out to all of you. I'd love to hear from you on how limiting that is today in terms of using these different modalities, how you think about it, and how you see that evolving or changing.
This is a really important point. I think that one of the most important differentiators between different treatment modalities today all has to do with delivery. There are some drugs that work beautifully when you're spritzing them onto individual cells in the laboratory. But then when you inject it IV or into the spine or into the brain, it only goes into a small percentage of the cells and has that magical effect that you can measure in the lab. And the number one differentiator between these different modalities is the extent to which they have been proven with a track record in different organs. I'm a neurologist, so I focus a lot on what is currently working in the brain. And there are a number of technologies that work really well in human brain that we've focused on, antisense oligonucleotides being one of those. And there are other amazing technologies that aren't quite there yet, that they just haven't been shown to be able to get into all the parts of the brain that we want them to. And that similar stories are true for liver-oriented diseases or bone marrow diseases, for heart diseases, for muscle diseases. And a lot of the brochures that you'll see about different modalities actually talk about the specific intricacies of how they work and how elegant they are. And that's important, but not nearly as important as delivery. And so really what's most important is knowing for each modality, where does it go and how well tolerated is it? Is it safe? Has it been shown to be safe?
I'm sitting here just nodding and smiling because it's all so true. With Wolfram syndrome, we sort of had to choose. This is just an example. For some diseases, one might be able to administer and it could go everywhere, or if you have the right vector, the right promoter, there are all these parts to delivering this stuff, which again, didn't exist 25, 30 years ago. in Wolfram syndrome, we had to choose, and based on what the patient said was most important, vision seemed most important, and it's actually a really good way to treat with a gene therapy because you have two eyes. And so we had to think about vector delivering to the eye, promoters, uptake, and then in Wolfram syndrome, you also have to think about where that's going to go and gets into the weeds. For each disease, again, the devil is in the details, but when you understand the biology, you can, and you understand what the patients really want, what they really need to really create better quality of life, then we can narrow it down. There are libraries of vectors now. There are libraries of promoters. And so what a lot of folks have to do is try this one, try that one, try that. They tried in the lab in an animal model and narrow it down. So that's what we've had to do for Wolfram syndrome, so that we have enough efficiency, meaning enough of the correct cells take it up to have the effect that we really want, which is to permanently fix those cells and prevent them from dying off.
And Daniel, how do you think about the delivery challenges?
Yeah, so delivery is critical for us. And again, going back to my two by two, when we think about probability of success is ability to deliver to the desired tissues. The big advantage of tRNAs is we don't need specific, we deliver right now using AV vectors. The great thing is we don't need specificity from the vector, but the engineered tRNAs are only going to read through the premature stop codon. It doesn't read a natural termination codons. So really the tRNA is what gives us specificity. What we really need is a vector that's going to get the tRNA to that place. And a lot of the work actually that we're doing right now is optimizing the construct so we can go at very low doses with AV, given all the issues that have been in the field with systemic dosing of AV. Our goal is to dose at go at very low doses. So that's where we increase efficiency.
We're going to take questions from the audience in a minute. So if you have questions or you're thinking of one, you could get it ready. But, you know, we're in this age of AI that's rapidly changing things. Tim, you talked about this almost plug and play model where, you know, if you could hit this one target in the liver, why not just changing the code and hit the one next over to it? But I'm curious if any of you using AI today integrated into your work and how you see that evolving and changing what's possible in terms of therapeutic modalities.
I think that AI is going to be incredibly important for accelerating the design of all of these therapies. I do think that pragmatically, if there is a AI magic bullet, if you think that there's an AI magic bullet out there right now for anything, any specific application, it's not there yet. It's vaporware, I have to say. I think that AI thrives off of having importantly well-curated and rich data sets. And I do think that there is sufficient data around how to diagnose patients and how to interpret genetic findings such that we're going to see AI being able to draft off of that and learn to make the delivery of genomic diagnoses more efficient. I do think that in this more rarefied world of therapeutic development, we have a long way to go before we're going to be able to make the AI designed designer molecule just like that. I'd like to believe that we need to prepare for that future and break down the silos that keep therapeutic development data cordoned off so that we can enable that to happen. That's actually one of my own personal big driving academic missions. But also just speaking to a room of folks who are thinking about what can we do now, I just want to be realistic. It's pragmatically, it's not quite there.
Are you looking at screening possible drugs for repurposing or?
So I love this question because 8 years ago, a Wolfram mom and I put our heads together. She's a molecular biologist. And we said, I wonder what we could get from Amazon that might help our kid. I mean, we've all thought that, right? And don't do that. But we just started talking because, again, as a pediatrician, I'm thinking, what's the basic problem? what are the symptoms? What's happening out here? What we did understand was that our protein had some issues. When it's mutated, it doesn't work well. We knew a few of its jobs, and we started thinking, we can't build, we've tried, we can't build yet a compound that will physically support it and nudge it into the right position. But we thought, how could we support it and maybe do some surrogate activity around the outside, right? And just help with the jobs it's not doing well. And we got lucky because after writing some emails and a couple phone calls, we did end up with a drug that was already being used for something else that had two such compounds working synergistically that it looks like so far, I'm going to say the data is promising. I get very excited about stuff, but researchers do not. The data is very promising looking. So we did use AI in a sense then. That was 8 years ago, so it wasn't real AI, but it was a Google search, to go through. Now we're actually using AI to do that on a much larger scale, to do a modified high throughput screen in Wolfram zebrafish. So you can do this safely, effectively, if you have the reagents to do good testing with good protocols and you've got your researcher. Yes, it can be used from that end to work backward and say, how can I work around the problem to support what should be happening in the cell for some diseases?
I thought you were setting up an Amazon Web Services. Daniel, is there any way you're applying it today?
Yeah, so we've been building capabilities, but I think like Tim said, it's not a silver bullet, and you need to build the data around it. At the end, it's data-wise garbage in, garbage out. You need to have the right data. So in our case, at Therapeutic, 77 nucleotides. And one change makes a tremendous impact on the read-through ability. And we do large screens. We screen 60,000 RNAs with single modifications. And eventually, the idea is to do that through AI, that we don't have to do a wet lab screen.
So we do have a few minutes for questions. If anyone has questions in the audience.
So question for Daniel. So as you think about, like, I guess maybe potentially using AI or other means, like, are there ways that you're also exploring of when you have certain therapeutics, where there are other commonalities, what other rare syndromes may benefit from these? Like, are there ways that you're proactively trying to find other groups?
Yeah, so again, when we prioritize the indications that we're going to work on, we look at size of the population, incidence of nonsense mutations, existing treatments, potential to be transformational, and can we deliver to that tissue? We start building groups. Our idea is to do a basket clinical trial or have one approval and then doing rapid label expansion. So if you think about it, one initial trial would be for these developmental and epileptic encephalopathies caused by nonsense mutations. And the idea is that anybody with seizures and delays and with a nonsense mutation could participate if it's the same nonsense mutation. Same thing for cardiomyopathies, same thing for muscular dystrophies. So yeah, at the end, we look at common group of indications, and the commonality has to be the endpoint that we're going to be looking at a clinical trial. And do we use the same vector to deliver and same route of administration to deliver to that disease?
I think there was a question over here.
Hi, thank you. I'm wondering, when we have diseases that have a variety of different kinds of variants across multiple genes, where you find the starting place? How do you know which gene to start with, what type of variant, where it all begins?
So go a little more into that for me, please. So is there a specific disease that you have in mind, or?
Yeah, so personally, I have classical Ehlers-Danlos syndrome. Most of the time, it's a variant on one of two genes affecting type 5 collagen, but we have a lot of different variants that are all individually rare. Like mine's been seen in about 30 people, and we have a lot of different ones across these two genes, sometimes a third gene.
Yeah, so excellent question. We have the same thing in Wolfram syndrome, hundreds of different mutations. Again, you have to get into the details, the weeds, and understand more about where do those mutations lie? Do they all happen to lie in one exon by any chance? Are they mostly missense mutations, single nucleotide nonsense mutations? Can you sort of qualify or quantify the types of mutations and then the type of tissue that you want to go to? So looking at each of those details, it's almost like there could be a checkbox roadmap just for this alone that probably is more complicated than the one we saw earlier. But your disease will have these things in common with other diseases. So making the connection to a disease that has a similar mutation profile, it may not be anything similar in terms of symptoms, but it may have a similar mutation profile. And then we look at how are they solving that problem. That's sort of a general look at that, but we can talk a little bit more about that too, or anybody else.
Actually, you could corral all these folks at the lunch. There'll be expert tables and you're welcome to choose one to sit with. We have time for one last question.
Yeah, hi, Robert Green, medical geneticist here in Boston. I've been leading the Baby-Seq project, which is sequencing apparently healthy newborns in order to determine the risks of genetic disease early. And as Tim knows, because he was part of that back in the day, you guys have spoken so much about this indirectly. Sonia talking about treat or prevent. Sarah, you're talking about treating all the variable onsets and even individuals as late as 71 in whom the disease was hidden. And Tim talking about the earliest possible diagnosis. If there's time, and I know it's a big separate topic, but could you zero in on the ways in which different therapies could be used early if there were pre-existing risk information after sequencing apparently healthy newborn babies?
I think that the really critical question when you have this opportunity to realize really quickly that a newborn, for instance, is predisposed to a condition, that's when you have the most opportunity. The types of interventions that we're talking about, and the types of approaches are all likely to be maximally effective in that early period. When for disorders that affect the brain, many of them affect the way that the brain functions and matures. And as we know, when we're born, there is so much important brain development that occurs that you need to set those patterns straight from the beginning to have maximal effect. I think that every single modality that we're describing here is most effective in those earliest stages. It's even more fascinating to think about a future in which you could intervene for that individual with a drug that doesn't necessarily even exist yet at the time that that diagnosis was conferred. Can we shorten the drug development process for some of these modalities down to the point where it doesn't have to be pre-existing on the shelf, but you can actually design it and then test it and then manufacture it and deliver it just in time for that individual? Granted, we're talking about a vision for the future that may be 50 years or 15 years or 5 years ahead. But that's really kind of what is driving the important efforts of Baby-Seq and finding and nipping as many of these in the bud as possible.
So at the risk of getting yelled at by the organizers, I'm going to ask each of you very quickly to offer one takeaway as we wrap. Tim?
I think my takeaway would be to sit down and confer with your scientists and your biotech and business advisors for each of your groups, and to take a hard, practical, cold, and dispassionate look at all the options available, and divide what you hear about into what can I do now, and what can I do in the medium term, and what are the modalities that I want to invest in for the future. Those that aren't necessarily yet proven today, but you think may be your main play in, say, 5 years, as opposed to next year. I think that going through that process as a discipline is really important to all the communities here. It's been the way that many of the most successful and most well-known disease communities have been successful in the past.
I'm going to echo that a little bit. Something we really like to say is get everybody around the conference table, and nobody can be alone at the center of the wheel. Everybody's going to have to be there together. And I will tell you, one of the best ways we found to do this was to have a research conference with foundations and researchers all in the room, because they present their research, you know the modalities, you know what needs to be worked on. You sit at coffee breaks and brainstorm. You sit at meals and brainstorm. It sounds impossible. The idea of setting up a conference to me was, oh my gosh, no way. It can be done. And I would say it was the most effective, efficient way to get everybody around the conference table collaborating and to know everything that was out there and what needed to be done next.
Daniel, you got the last word.
So I'll give a short story with takeaway A and B, but quick. So when my daughter got diagnosed 15 years ago, we're the doctor, he gives us a diagnosis. And of course, I'm an engineer. So I said, okay, she has epilepsy Dravet, let's cure it. And he's like, oh, no, sorry, there's nothing you can do about it. So for me, that was a shock. So my first message is there's always something you can do about it. And I'm probably preaching to the choir here. All of you are doing something. So find what's needed in the field, you think it's possible, what you're good at, what you're passionate about, and what's going to keep you going. I mean, if something gives me hope, me and my family, about the whole situation and my daughter, is that we're doing something about it. So that's critical. So my follow-up question to the doctor is, well, but I've heard about gene therapy. He's like, oh no, that's science fiction, not in our lifetime. 15 years later, it's a reality. Thank you to people like Tim and others as well. So there is hope, and the time is definitely now. So keep pushing that there are definitely therapies that are transformational for everybody's kids.
So you are all welcome to continue this conversation at lunch. Please join me in thanking this extraordinary panel. Tim Yu, Sarah Gladstone, Daniel Fischer. Thank you.